So I will speak for Osiris regarding particularly studies on biosciences by Quens. And sorry, there is, okay. It's just that they are a bit slow. So Osiris, as you all know, is a time-of-flight backscattering spectrometer, is a low-energy spectrometer for quasi-elastic and inelastic neutron studies, and it is used to study relatively slow motions, so diffusion, rotations, translations, or vibrations. Studies on a very wide range of themes have been going on in Osiris, like catalysis, hydrogen storage, advanced materials, hard and soft condensed matter, including biological systems. And I, for time's sake, I am always only showing you some chosen examples. Some of those were already shown last week, but for those who were not here, so studies on sodium-ion batteries, for instance, as attractive substitutes to lithium-ion batteries, studies on highly efficient solar cells, on catalysts, also now going more to the biosciences side on intracellular water in extremophiles, also in human cells that I will show in a while, characterization of water dynamics on the surface of proteins, which, as we heard just now, is so important for functionality, controlling cell membranes, molecular insights into the toxicity of certain compounds like ionic liquids to mammalian cells, and very importantly, some simultaneous experiments like this new capability with Quens of simultaneously performing DSC and Quens measurements, which is really a very nice thing for a great number of systems. So the Endeavour upgrade for Osiris guide system will achieve an intensity gain factor of around 5 at the sample position with understandable and very desirable benefits for analyzing biological samples, which are, as we all know, highly heterogeneous, so very complex systems with a very large mixture of very heterogeneous molecules, and usually we have them in small amounts. So the intensity gain is really important in this field. So from the Portuguese neutron users community, our group is the molecular physical chemistry group that comes from the old University of Coimbra, which involves, in these neutron studies, involves a team of about 5 or 6 people, including myself. We've been using ISIS for about 20 years now, very happily, I should say. And the first thing I will tell you about is the first neutron scattering studies that we performed in the last 5-6 years on two systems. Firstly, on normal to cancer transition. So we have investigated human cancer versus human healthy cells. And very recently, in fact, in June, we were the first foreign users that were able to go to ISIS. And we were very happy to be there and work there again, although with a mask, on human cancer biopsies. So to try and understand the malignant transition, so to non-malignant to malignant transition. Also, we have studies already published on intracellular water, which can be considered as a potentially very important drug target. And within this subject, we studied platinum and palladium drugs, which are new drugs that are developed in our group, their impact on human cancer cells. So again, always human samples. So regarding normal to cancer transition, although it is a very high impact on human health, it's still a very ill understood process. We know that it involves biochemical and genetical changes, but the role of cellular dynamics should not be disregarded because it's really important, since it is closely associated to plasticity, either of cells or of tissue matrices. It regulates malignancy and invasiveness, So it can be taken as a hallmark of cancer and metastatic ability. So the cancer onset can be seen as a dynamical phase transition, in fact. And the studies that we performed in cancer cells, either versus non-malignant cells, so healthy versus cancer cells, could show, as you can see here, for different types of cancer, breast cancer, the triple negative, the metastatic breast cancer, and the refractory, so hormone-independent prostate cancer, we could very clearly see that there is an increased flexibility for the malignant cells if you compare the healthy in black with the cancer cells in red. So there is a different dynamics. Also, these different dynamics, this increased plasticity depends very, very strictly on the type of cancer. If you look at this image, you can see that, for instance, for osteosarcoma, which is bone cancer, the plasticity is lower than for prostate cancer. So this depends a lot on the histology, on the biochemical and dynamical behavior of the type of cancer. So very recently, as I told you, we probed human cancer biopsies that we collected with patient consent at the Institute of Oncology of Coimbra, with which we have a protocol, and we compared them with non-malignant tissue, which are the surgical margins that are removed during a tumor removal operation, surgery. And also, if you compare, first of all, the breast cancer tissue, so this was performed for breast and for tongue cancer. If you compare the tissue with the cells for a very similar histology, so the same type of disease, you can clearly see that there is a higher mobility for tissues versus cells. And we should not forget that these dynamics, these Quenz measures dynamics, are mainly due to water mobility. So water dynamics, water diffusion is the main contributor to these Quenz profiles. Now, if you compare the two types of cancer that we probed, so the biopsies for breast cancer, the margins for breast healthy, and the same type of samples for tongue squamous cancer, you can clearly see a very interesting result, in my opinion, which is there is definitely a different mobility for malignant and non-malignant tissues. But this difference depends on the type of cancer. And in this case, you can see that while for tongue tissue, the healthy tissue is more mobile than the cancer one, the opposite occurs for breast. Also for the tongue tissue, the difference is larger. So this is already an interesting result that, of course, these are really fresh from the oven results. So these have 3 weeks. Of course, we are in the process of analyzing them. So if you look at these profiles and these elastic window scans, you can clearly see this different dynamical behavior for the distinct types of cancer if you're comparing breast to tongue cancer. So breast being more flexible. Now going to the anti-cancer drugs, so new targets are needed for chemotherapy. So the conventional targets, which are usually an enzyme, a very conventional target being DNA, as it's depicted in this slide. So these are not enough, and new targets are needed. Also, a multi-target approach is needed. So it would be wonderful if, for instance, metal-based drugs like cisplatin, for instance, could at the same time affect DNA and other targets simultaneously. This would greatly increase the efficiency of the drug. So here we compare cisplatin, which is a conventional drug used in oncology since the 70s, with a newly developed drug in our lab, which has, instead of one metal center, has 2 metal centers. So the difference is immediately obvious. While cisplatin can only bind to 2 places in the DNA strand, and usually through intrastrand short distance adducts, our drug can bind to 4 different places, so 1, 2, 3, 4, because the chlorides are hydrolyzed, to more places in the DNA helix. Since the metal centers are separated from each other, it can yield interchain adducts at a long distance. This leads to a more severe damage of DNA. So in simple words, this is more damaging. It will cause, theoretically at least, more cell death, which is what we need. But why not looking at intracellular water as a possible drug target? So intracellular water, for instance, if you think of the cytoplasm, is in a cartoon-like way, just like a highly crowded intracellular medium, just like a highly crowded swimming pool. Any change in the water of this swimming pool will for sure affect each one of the persons that is inside that swimming pool. So within a cell, when we disrupt the coherent dynamics and structure of cellular water, we will probably, very probably, disturb the normal cellular function, because water affects the function of the biomolecules. So this can lead to interference with cell growth and eventually even cell death. So if a drug affects, apart from DNA or a specific protein or a membrane, if it also affects intracellular water, then this can be another very important target, mediating its activity. So going from the cell culture lab to the beam, which is what we intended to do about 5 years ago, in order to probe this hypothesis, is far from being a straightforward task, as you can all imagine. So you have to have a cell culture lab, which in those days was still in the research complex, so far away from the hall 1 of ISIS. So we had to grow a huge amount of cells. Just for you to have an idea, we had around 100 to 120 culture flasks like these, which are 75 mil. So we have to grow a high amount of cells in order to have a large amount of pellet to put in the beam. And then, of course, we had to add our drugs and be sure that we had beam at the time points that we wanted to study, 24 hours, 48 hours, 72 hours. The beam could not be off. It should not be off. Otherwise, the time point would not be 48 hours, but it would be much more. So it's far from being a straightforward task. Anyway, we accepted this challenging experiment. ISIS accepted our challenge, and this was successful and was in fact the first study in human cells to be performed by neutrons. And we found some very interesting conclusions. So within a cell, you have two types of water. You have the cytoplasmic water, which is more flexible, like the water in the swimming pool. But then you have a highly ordered water around the biomolecules, around DNA, around the protein. This is a very organized water, 1 or 2 or 3 layers of water, which are less flexible than cytoplasmic water, which is usually has a bulk-like dynamics. And we saw a very interesting thing, that for hydration water and intracellular water, the drugs had opposite effects. While it induced an increased flexibility in the hydration water, so hydration water was disorganized in the presence of the drug, it was rendered more mobile. For the cytoplasm, the contrary occurred. There was a drug-induced restricted mobility. So the cytoplasm became more rigid in the presence of the drug. So this was really an interesting effect.
Maria, sorry to interrupt, just about 1 minute left, please.
Okay, and if you look at it in a cartoon-like manner, you can see this drug-prompted concentration-dependent restricted mobility in the cytoplasmic water, while you have the opposite effect on the hydration layer. So for DNA hydration layer, for instance, this obeys to a jump diffusion model, and there is a drug-prompted mobility increase. So these are a decreased organization of this hydration layer. This was interestingly also different for palladium versus platinum drugs, which had different effects on the water or on the biomolecule itself. So for time's sake, I cannot go into details, but this was an interesting result as well. So overall, the Endeavour program, and in particularly the upgrade of the Osiris spectrometer, will benefit studies on biosystems in cells, in tissues, also because it will allow us to have smaller amounts of sample. It will allow us to acquire data in shorter times, so more data, more results, both for improved anti-cancer drugs, so aiming at a better chemotherapy prognosis, and also trying to understand the normal to malignant transition, which will lead us to better and earlier cancer diagnosis methods. So this was what I wanted to tell you about Osiris, the new upgrade within Endeavour, and also our latest results on biosystems. Thank you.